the Fireside chat. We have Jonathan Cook here, Research Associate at Jesus College, among other things. And then, of course, Sandy Douglas, who's going to share about scaling the Oxford COVID vaccine. We're going to have 45 minutes in that and then merge into a bigger panel. I think, Jonathan, I'll yield it to you. And looking forward to this. I think so, yeah. Tracy?
Hear me? Amazing.
Can you hear me as well?
Yep. Fantastic. I guess everyone is trickling in, so that's great. Thanks so much for joining us. Again, as Jakob said, we've got Professor Sandy Douglas here to chat to us a little bit about, I guess, how we scaled the manufacturing of the Oxford COVID vaccine. I don't know if that was your quote or if that was Jakob's. No. So, but to start, I think it would be great if I could get just a little bit of a chat about sort of what happened back in 2020.
Okay, that could take 45 minutes. I guess I should start with a disclaimer. So I'm not trained as an engineer at all. Up to February 2020, I'd thought about scale up roughly this much. I trained as a medic. I did a PhD on malaria vaccine development, and then I'd started work on early phase clinical development of a rabies vaccine using the adenovirus technology that was also subsequently used for the Oxford COVID vaccine. And I made the mistake of trying to make my grant application a bit sexier by saying we'd do something a bit new with this adenovirus tech. So we'd done various different diseases with it before, and I thought the MRC wanted something a bit new. So I said I'd thermostabilize it using a pretty unproven tech that someone had done a PhD on in the lab, and it looked okay. What I learned from that is that translating things from promising PhD projects into GMP, good manufacturing practice, compliant, regulator acceptable clinical processes is very hard. We never managed to make that work. But we were very lucky that a byproduct of that was that we needed to make a bit more adenovirus in a different buffer and at a higher concentration than the university had made before. Previously, the university had made adenoviruses in a totally non-scalable but very safe process to do small clinical trials many times, but it was really not a method that could go beyond a few hundred doses. So what I had been forced to develop for my rabies project and all the troubles it had was a method that was in principle scalable, and we made it as simple as I possibly could so that our own in-house manufacturing facility could actually execute it. And I was thinking, well, what might this be useful for? And I thought, well, possibly something that's very simple and works for a platform vaccine technology, something that can be applied to lots of different diseases, might be useful for outbreaks of new diseases, and it might be useful for transferring that process to different places around the world. So that's where I was in January 2020. And I'd put out a paper in November 2019, and I was sort of spinning it. I don't know who submitted papers, and you try and make it sound interesting to the editor. I said, this could be useful for outbreaks. And then I wasn't really expecting to do it. I was telling Jonathan, I had a big grant application, which I submitted about February the 4th. I went away for a nice weekend with my now wife. Sunday morning in bed, read the New York Times, and I saw a graph of R0, estimates of R0 versus case fatality rate for this coronavirus. And it was extremely obvious, in my view, to anyone who'd done Epidemiology 101, literally Epidemiology 101, that this was worse than anything that the world had dealt with since the Spanish flu, and it was going to be the worst global emergency since the Second World War. So that sort of spoiled my weekend away somewhat. I spent the next 10 days thinking, what on earth do I do? Because I wasn't, I'd never worked on respiratory viruses before. Wasn't my thing. I spent a bit of time sort of agitating that we should be moving faster. And then I realized that, well, actually, manufacturing was probably the university's biggest weakness. We had a vaccine platform technology which was suitable, that had shown potential for other coronaviruses before. I was pretty optimistic that it would protect against this coronavirus as well. We had the capacity to do all the preclinical trials, early phase trials, and right through to phase 3. You know, the university has a very broad capability. We had in-house clinical grade manufacturing, which is a very unusual capability for an academic center at small scale. But we had absolutely no experience or credibility in large-scale manufacturing. And it was fairly apparent that if you actually wanted a vaccine to have impact in the world, you were going to need a large amount of it, and you were going to need the credibility that you could make a large amount of it in order to get industry partners. So I set about working on that. Initially, we did, I don't know who's been on a resuscitation training course, and you're taught the first thing you do when you encounter this situation you can't deal with wholly on your own is you call for help. Yeah, so we did that. We sent out an email to the UK Bioindustry Association, the sort of trade lobby group, saying we've got this promising vaccine, we need some help to make it. And from there, I ended up leading a consortium of different manufacturing companies working on the scale-up. We went from, within my group, I had literally one postdoc working on this stuff before the pandemic, another one who was on maternity leave. So we went from that to some hundreds of people working on it across 2 contract manufacturing organizations in the UK, one in the Netherlands, one in China, and one in India by March 2020. We had no money through March 2020, so we could maybe talk about that if you're interested. And then in April 2020, serious backing arrived from the UK government, and we had negotiations with big pharma partners. So short answer is I developed a scalable process for making this thing. We had to figure out how to show we could go from benchtop scale to plausibly commercial scale. So we did that very rapidly with this consortium, and then we used that to help us form partnership with ultimately AZ.
So, I mean, that's January to April, 120 days or something. Fantastic work, but we were discussing this a little bit earlier. We have all these great minds at Oxford, but there's great people all around the world. There's also people who manufacture vaccines for a living, where Oxford was having a manufacturing setup. What do you think, what do you attribute sort of this particular
so I think that's a really interesting question. So I'm obviously lucky to work with some great people, and we had fantastic collaborators outside the university as well, and that was obviously essential. But as you said, various other organizations had very good people and actually much more experience of doing this sort of stuff than we did. So I think what made the difference, a number of different elements. So we had a combination of control and ambition. So the university controlled all the IP relating to our product. So we could basically do what we wanted. And there was a clearly exceptionally unusual business opportunity, to be blunt. Everyone wanted to do the right thing and help, but also they could see that if you were first, that there was probably quite a lot of money to be made as well. So lots of people wanted to help. We were very lucky with that. But we also had ambition. Most of the leaders of the program had cut our teeth, I suppose, working on vaccines for global health, malaria, TB, typhoid, that sort of thing. And so we were ambitious for, our aim was public health impact rather than money. So that made a real difference in how we approached it, and in particular on the manufacturing side, the priority we put on low and middle income country supply, which we could talk about. But I think if you think about the organizational structure, I think one of the really unusual things about the university is that it's a big organization with huge capability under its roof. There are some things that big farmers can do that we can't, obviously, but many of the things they do, we can do. But within that, certainly within our department, it's actually quite small teams working on individual products. Very few people have permanent jobs. I don't have a permanent job. I have to get grants to pay my salary. And so those of us leading those teams are essentially running our own little small businesses. And whatever product it is that we're developing, we have to solve all the problems because nobody else is going to solve them for us. Obviously, we try and collaborate, but at the end of the day, it's us that cares about our own products. And so we had a handful of people who were generalist leaders. And I think that was very important because other places had deep specialist expertise. But I think in many places, the manufacturing people thought that the clinical trials would be unavoidably slow. The clinical trials people thought the manufacturing would be unavoidably slow. And you had to understand both to identify the opportunities to go fast. And then also from that sort of small business experience, I suppose, and the global health ethos, there was a degree of experience of negotiating with companies and trying to take something which initially wasn't, if you're developing an early phase malaria vaccine, it's not commercially attractive, but at a certain point it becomes investable. So we had between us a reasonable amount of experience in how to form mutually beneficial partnerships with companies in this sort of area.
I think a couple of the things that you touched on there, it sounded a lot like you had to break some rules, or at least go around, the established norms. So, for instance, you're describing the rapidity of clinical trials work, and knowing that waiting a year for a phase 1 to be done was not going to work in this situation. But you yourself were saying you were just starting out, right? You were applying for grants to start up your group. You had one postdoc working. So you're fairly junior at the time, and you were sort of reinventing what rules you wanted to follow. Is there anything that you can tell to this I think when I look around, everyone looks pretty young and fresh-faced. Some advice to them about that kind of outside-the-box thinking, or?
I don't think that there were 6 principal investigator leaders of the program within Oxford, of whom I was the most junior. I wasn't quite at the stage of just having one person in my team. I had one person in my team working on this, but I had several working on other things as well. But I was definitely the most junior person in the room, and perhaps the only advantage really that brought was the naivety to think we could do things that were ridiculous, or that other people thought were ridiculous. We didn't break any rules. We didn't break any rules at all. But we did have a lot of dialogue with regulators about what was an appropriate way to implement the rules in this, unprecedented situation. So it's an accepted principle, certainly for medicines regulation in the UK, that the MHRA, the regulator, has a mandate not just to promote patient well-being by ensuring safe and efficacious medicines, but they do also have a mandate to not block and hopefully support innovation. And what that means in practice is that if you want to do things in a new way, that you believe and you can produce evidence is as safe as the established way, you can go and you can pitch it to them and say, this is how I want to do it and this is why I think it's okay, what do you think? And so it was that very established process that we used, just with everything moving very, very fast, because we had a hotline and they'd consider things essentially in real time.
But from what I recall reading, it seems that you were already organizing all of this infrastructure and these manufacturing processes to make large-scale vaccines for a vaccine that had not yet been proven to work.
Yeah. Yeah. So I guess among the things I was doing in February 2020, partly coming out of the stage of figuring out, heck, what on earth do I, Sandy, do about this thing, was some sort of advocacy work, because it was, I guess, alarming initially, the idea that if we knew we could make a few hundred doses in-house, a couple of thousand doses with a smallish partner we'd worked before in Italy, and with those we could plausibly get to an efficacy result. And if the government hadn't locked down, we could have had an efficacy result in May 2020. If there'd been lots of cases of coronavirus in the UK in May 2020, we'd have known it worked sooner. And so I was thinking, oh dear, so maybe I'm going to end up responsible for this manufacturing, and we're going to have this vaccine that we know works, but we're not going to have any of it to give to anybody. That's going to be a problem. And that also applies to other people's vaccine programs as well. And we were desperately short of money at that point. So I did a little bit of work. My wife's an economist. Her boss was the former chief economist at the Department for International Development, who was one of our sort of relatively few connections into UK government at that point. And we together wrote a sort of, basically a position paper that said, tried to make UK government and anyone else that would read it aware that it would be there was a need, if we wanted vaccines at a global level as quickly as possible, we needed to invest in manufacturing at scale, at financial risk, before the efficacy results came available, and we needed to do that for a portfolio of different vaccine candidates, because any one of them might fail, and that would probably cost something like 100 million per candidate, but at a global level, when you consider the costs that this epidemic was going to have, that was trivial, so let's get on and do it. So we started making that case very early on, and I think Patrick Vallance, the UK Chief Scientific Officer, was pretty early on to that thinking as well. The UK Bioindustry Association was pretty quick on that thinking too. So yeah,
right.
But by April 2020, UK government had got that idea, and it had set up the Vaccines Task Force under Kate Bingham's leadership, and that sort of venture capital model of trying to make smart choices in what to invest in across different programs and building a portfolio. That was exactly the right approach, and manufacturing was a big part of that from the beginning. By May 2020, other governments had recognized, and they, so suddenly there was, a big influx of money, and it was very much de-risked.
Now, remind me, at that time, was there a chimpanzee adenovirus-based vaccine that had been made at this scale?
No.
No.
So the biggest scale at which other adenovirus-vector vaccines had been made was J&J's. J&J had invested very heavily in the platform, not a chimpanzee adeno, but another one. Very similar manufacturing process. And they had made, I think, hundreds of thousands of doses, and they got through to licensure. for their Ebola vaccine. So there was proof of concept out there that it was possible to make this sort of vaccine. There were, I think, interesting differences between their very sophisticated approach and ours, which was much, some might say simpler, some might say cruder. If you had asked me at the beginning of the pandemic who's going to make more vaccine, I mean, I'd have been, you'd have to be mad not to say J&J. Unless, I guess, niggling at the back of my head and many colleagues' heads was the idea that Big Pharma is pretty slow at doing many things, and they might just fall over themselves. And so I started thinking in sort of hundreds of thousands of doses scale in February 2020. So sort of similar, I suppose, to what J&J had done before. I got a little bit of funding from the UK government saying, hey, if we make a batch that sort of scale, we could vaccinate all the doctors or all the nurses, that sort of thing in the UK. In early March, I had a spreadsheet I didn't show anyone that showed, it was called billion dose scale. It was just some very crude Excel-based maths of like, what are we going to run out of if we try and do this at really big scale? Where's the bottleneck going to be? And at that stage, I was too embarrassed really to show that to anyone else, and I would have, I wouldn't have believed that what actually happened would happen, which was that we actually, by the end of 2021, the period of sort of peak emergency rollout, about 2.5 billion doses of ours had been made, and J&J about 500 million, which was still big and important, but quite a difference.
So what was it on that billion dose spreadsheet that you were worried we were going to run out of? Is it?
So I spoke to sort of suppliers of various reagents, and they said, yeah, we can make quite a lot of that, don't worry about it. One or two of the things people had told me I should worry about. The bottleneck, it was fairly obvious, was simply litres of bioreactor capacity, because we could make about 1,000, 2,000 doses per litre in a process that took about 6 or 7 days. So, you do the maths. It was obvious if you wanted to make 1 billion doses, you needed tens of thousands of litres of bioreactor capacity. And there was no single facility globally that had that. So we needed multiple facilities. So through necessity, we went for a scale-out model where we went to multiple different sites simultaneously. It did happen. One of them, the Serum Institute of India, had an extraordinarily well-suited facility that they developed to make monoclonal antibodies, but just happened to be a really good fit for our process at really big scale, because the Serum Institute of India is the world's biggest vaccine manufacturer, not by financial volume, but by number of doses. They make huge numbers of doses. So they ended up actually making more than half of the global supply on their own, but it was the scale out that allowed us to get to the total.
And so in moving from something like a scale up, where you've sort of reached your limit from the technology standpoint, and now you have to scale out, is there anything that you had to keep in mind for sort of QC or quality control between the various manufacturers on different continents throughout the world?
I suppose we haven't actually talked about the technical scale-up bit at all. Maybe we should come back to that.
Yeah, fair enough.
But it was a question that everyone was asking us around April 2020, because we had this scale-out consortium. We didn't yet have, we had the Serum Institute of India were on board, but some of the, top brass in the university, I think correctly, felt that we needed a Western multinational pharma partner. And we were being asked repeatedly, you've got this higgledy-piggledy consortium of different manufacturers. Pharma's going to hate this because it's going to be different product from every different site. So we were trying to provide reassurance on that long before we'd actually made anything. So the fact we had the IP control and the business opportunity was massive meant that all the different members of the consortium were strongly incentivized to truly cooperate in a way that they normally don't, you know? They'd normally be competitors. And many people said they loved working on the program partly for that reason. But across all the different sites, from the beginning, we had common standard operating procedures, common input raw materials. We sort of bought the global supply of new bioreactors from Paul Biotech at one point to try and, they all had certain bioreactors, but we wanted them all to have the same one. We centralized the analytics in shared labs, in a shared lab. Ultimately, though, I think it was important that we did have a single coordinating entity with the experience of managing massive scale manufacturing in AZ. I suppose it was also important actually that they didn't have another vaccine program on that scale of their own, so they actually had no choice other than to stick with our scale out model because they didn't have a facility of their own that could do it. But they did have the know-how to provide a coordinating umbrella to basically try and force all the different sites and provide the oversight to make all the different sites put out product of the same quality. Even with that, there were challenges in achieving that. It was hard.
And I guess at the other end of it, so now you've made 1 billion vaccines. Did you have any hand in the distribution side?
No, thankfully. So the important thing about the distribution for our vaccine was that it was suitable for standard, fridge refrigerated, not frozen like the mRNA vaccines. It was the fridge distribution was important, especially in low and middle income countries. We'd done a bit of work on temperature stability stemming from my rabies program. But really that distribution was handled by AZ and also by Serum Institute of India. So the fact that Serum Institute of India remained part of the program was really, really important. And it was quite an unusual process, the negotiation that led to a partnership with Western Pharma, but actually them being more or less forced by the university to keep on working with the Serum Institute of India. And that came back to the IP control that the university had, plus the fact the UK government had made significant investment and there was a sort of international aid voice in the room. Plus the fact that under the unusual emergency situation, the big pharma partner really needed a small number of individuals in the university to be happy and play ball. So yeah, that's how we kept Serum Institute on board, and that really was the biggest single thing that achieved the global distribution.
Wow. So it sounds like this was, a lot of elements came together just so to make this possible to happen. Was that planning, or was he's shaking his head? So do you think we could do it again?
So it definitely wasn't planning. The idea that one might plan ahead of an outbreak was definitely out there. The Coalition for Epidemic Preparedness Initiative, CEPI, had been set up as an international funder by Gates, Wellcome, some governments after the 2014-16 Ebola outbreak in West Africa to try and do exactly this sort of planning. One or two people, a guy in Queensland I'd seen give a great talk about this and invited him because I thought nobody else was doing it pre-pandemic. But we certainly did not have a well-figured-out plan of how this is how we do it in an emergency. And to be honest, even CEPI, they were really thinking about things on the scale of that West African Ebola outbreak, which is very different from making billions of doses and global lockdowns. So nobody had a plan. And actually, it was one of the important and deeply, deeply scary moments of the whole thing was realizing that nobody had a plan, there were no grown-ups in the room, we're going to have to figure this out. Which was pretty horrible, you know, at a point when Boris Johnson was still showing off, shaking hands with people at rugby matches and so on.
But for your group now, moving forward...
Do we have a plan now? No. We would love to have a plan now. We have a better plan now, but I'm afraid that we don't have plans that are as good as they should be. So CEPI is largely focused on low and middle income country preparedness. And politically, there's been a big push arising from the shortage of vaccines in African countries and the realization that there wasn't African vaccine manufacturing capacity to establish that. So that sort of huge amounts of effort are going into that. Personally, I think that's wrong. It's a contentious point of view, but I think we should be investing to make it incredibly easy and cheap for places like Serum Institute of India that already have massive scale to turn their production lines over and make enough vaccine to saturate the world within a couple of months in an emergency, and then it doesn't matter where it's made. Yeah? So I think there's been a really important strategic miscalculation by CEPI on that. So they're focusing mostly on LMICs. The UK, well, we all know what the US government's doing. The UK government, I think, has drawn the conclusion that we did okay in the pandemic and has become complacent. The EU has thought the opposite. They've thought, well, heck, we didn't really have any of our own vaccines. Not much came through, or at least BioNTech was an EU vaccine, but it was developed, with Pfizer. And supply in the EU was relatively slow, and so they have invested in a plan for large-scale manufacturing capacity. The problem is they don't have it connected to a plausible development pipeline of candidates, actually developers that would move quickly enough, in my view, to give them candidates to put in their manufacturing capacity. So in my view, unfortunately, we still don't have plans that are as good as they should be. Yeah, it's deeply frustrating. And, Bundy-Buggio's come along, and this, I think, is the third outbreak there's been that's prompted some emergency response from us. There's been another strain of Ebola and a Marburg-related virus over the past 3 or 4 years, where each time we've sort of scrambled to put something together. Each time there's phone calls with CEPI and the WHO and saying, how are we going to run a trial in a difficult part of Africa? In some respects, the planning has gone backwards rather than forwards. Sorry to be depressing. Technically, we're in a much better position.
So maybe we should switch gears to talk about the technical aspects of things. You described our local CBF, so this is our GMP facility that can create a couple of hundred doses that could, you could vaccinate 1,000 mice, or enough for a phase 1 trial. Were there any major hurdles going from that sort of scale into what you described, the 100,000, let's vaccinate doctors and nurses?
So it needed a completely different type of process. So the phase 1 trial batch was made using our in-house process, shake flask, and a floor-standing ultracentrifuge, a few hundred doses. Can't scale that up, really. The first phase 2/3 dose was made by this Italian company using methods that would, in principle, be scalable, but they just had an incredibly low-yielding process, bad cells. My group, we had this method that was, in principle, scalable. We'd made the rabies vaccine using it. My postdoc had come to me before Christmas 2019 saying she'd, changed the culture medium and added some feeding of the cells, and that had boosted the yield 5 times or so. And I'd said, okay, replicate it, please. And she came back in January 2020. Unfortunately, that did replicate. So we were literally at 30 milliliters in a shake flask with that higher yielding version of the process in January 2020. But we couldn't see a reason why it shouldn't scale, basically using the same bioreactor and purification process that we'd used for rabies. So we just stuck it straight into that at 3-liter scale, did some very simple stuff to see whether filters were going to clog up or that sort of thing. One of the companies that responded to that email to the Bioindustry Association that I mentioned offered to run 50 and 200-liter scale batches for us in their facility in Portsmouth, Pool Biotech. So we did simply linear scale up. We did nothing to actually figure out how to optimize the sizing of filters and so on. We just said, let's go with minimum possible risk to 200-liter scale, because that's what we need to show to persuade partners that we can make hundreds of thousands of doses and it's commercially investable. So by that time, the UK was in lockdown, but basically we did that as quickly as the cells would grow. So my project manager and a medical student went down the A34, him driving and her in the back of the car shaking the flask, to take them down to the pool facility. Grew to 50 litres, did a batch, grew to 200 litres, did a batch, and that was good enough to get AZ on board. And and there weren't really any big technical problems there, other than the fact that we noticed that one of the filtration steps in the purification was clearly not going to work at any larger scale. So that we then had to fix before we went on to 1,000+ litre scale.
So I know this is all really fascinating, and we've actually almost run out of time. Did you want to see if anyone has any questions from the audience that we can maybe do too?
Sure.
That's on you, yeah.
Yeah, thank you for sharing, for this session, very interesting. I have one question, which was bothering me for all these 6 years. So, in sense of I understand that, the process was much shorter than it's usually for any vaccine development, right? So some of the steps had to be skipped. I'm meaning safety checks. So, or how did you do all the safety checks? Not you, yeah.
So
How could you do that so short, the whole cycle? Not only the manufacturing, but the whole cycle of the vaccine development and approval. So that's my question.
So nothing was skipped. And the short answer to how it was done was money. So a lot of things that would normally be done sequentially, wait for the phase 1 result before you start scaling up your manufacturing, or before you put in place the plans for the phase 2, were all done in parallel. And so, by money plus the fact that we had prior experience of vaccinating hundreds of people with other vaccines based upon the same technology platform. So that gave us, we knew that there was shared safety across the platform. So that gave us the confidence to expand the trials to thousands of people by late May 2020. By the time the product was approved in December 2020, it had been in tens of thousands of people, and there were several months of follow-up on those tens of thousands of people. So there was a lot of safety data accumulated by that point, just as much as you would have for a vaccine under normal circumstances. On the manufacturing process side, there was process validation was done as it would be done normally. There was ongoing, process characterization, in parallel with the deployment, increasing depth of understanding of the platform, but nothing was skipped. As long as you've got the money, yeah, it's possible to make vaccines incredibly quickly if you've got the money. But the problem is that even in the early, if you think back to January 2020, it wasn't obvious that it was going to many people, that it was going to be an emergency on the scale it was. So there's always a period when, in my view, it's worthwhile as a global public good to be putting money into responding to a given outbreak, but it's not yet commercially rational for any individual company. So there's a really important question about how to make it as affordable as possible for response to be organized at a global level as early as possible in outbreaks so that we don't lose time. Because if you've lost time, you don't get it back.
Hi. Yeah, thank you. This is really interesting. I guess my question is, so much of it hinged on the platform having been tested before. So which other platforms are waiting that should be being tested now that we will come to recognize we rely on? Like, are there any that spring to mind?
I mean, mRNA is was the sort of dark horse. There were really, I suppose, 3.5 platform technologies at the start of the pandemic. There was DNA, plasmid-based vaccines, which had never really worked and didn't work. There was adenovirus. There was mRNA, which up to that point had not looked very good clinically, but there were, some encouraging preclinical signals. And the half is recombinant protein. The reason recombinant protein is a half is that every different recombinant protein behaves differently biochemically during manufacturing. And so it's not really possible to have a true platform manufacturing process formulation that works for everyone in the same way. mRNA now is obviously a mature platform. So, we have that. I think the thing we do not have that we should have is a platform that we have confidence will induce sterilizing mucosal immunity. For those that aren't into vaccines, what I mean is something that will stop you getting a mild COVID infection, sneezing it, and giving it to someone who's actually going to get sick. So we don't have that. We don't have... I think we would have a real problem if we had an outbreak with high mortality in young children, because we don't really have confidence in the adenovirus platform in young children. It would actually be easier in some ways in a high mortality context because a 1 in 50,000 risk. We know there's this blood clotting problem some of you may have heard of with the adeno platform. That's a problem in a situation where a person gets a small benefit from being vaccinated, but in a high mortality outbreak, you might not be so worried about it. mRNA, myocarditis is a thing, so children, I think, are still a bit tricky, but probably would be okay if we face something really bad. The thing we should have, but we don't, is the mucosal vaccines.
Okay. Andy, thank you so much for chatting with us. I think we're out of time. I bet that we'd be able to talk for another hour if we had it. But anyways, join me in thanking Sandy Douglas.
All right, thank you both. We'll be resetting and back at 16:20 with the panel for today.